Here's a very brief video to explain in a little more detail our assignment, homework assignment, to create a physical model of chick embryo based on this image. And the embryo will have labels on it or next to it like this, and it'll have most of these, but some of these terms like blood island we haven't learned about. So it doesn't have to have every single one of these, but it has most of them, and certainly the ones that we are learning about and focusing on. It also might have a couple more that you have learned about because we've been learning about the vertebrate phylotypic stage, and this is at the vertebrate phylotypic stage. What's the rationale for why we'd want to take time to build this model? One is this is what we looked like at the same stage of development. Birds, reptiles, mammals, they all look the same at this stage in development. Two, development proceeds in a direction, and as a result, we can see lots of different stuff happening in the same embryo at the same moment in time. And so there's lots of different stuff that can be represented that's happening. And then another reason is that model building is a very good exercise and very good activity to help you learn about the chick embryo and our bodies, and also learn about model building. Model building is a good skill to work on. Okay, so I'm going to put some building materials outside of my office, which is in room 476A. Oops, it's not going to be able to write with this right now. Sorry, folks. I'll go over to here. My office is room 476A, science building. And the materials are out there all the time, so you can combine any time. And there's some tables nearby, and you can work at those tables. And so you don't need to buy any materials. You could also come with classmates and build models together, and that's perfectly fine. Each person is going to turn in their own model, but you're welcome to cheat off each other and build models together. That works great and is a good activity. So as many people as you want can come together and just work on models together. If you have these kind of materials at home, you're welcome to build your stuff at home. I just don't want you to feel that you need to go out and buy anything. And it is good if you have stuff at home. It is good to have some colored paper and things like that, so you can really get some variety. Because again, one of the goals is to help you learn this material and think about it, and also have sort of a visual, tactile, physical, three-dimensional memory of these topics, because they're very important. Okay, and so that's the assignment, and then make it, and then just take some pictures and email them to me. You don't have to turn in the actual model, just take pictures of it, okay? Like that. I included this example of one that a student built a couple years ago, and I did that because it looks just about like the original drawing, and yours doesn't have to. I've seen a bunch of different kinds of models based on this assignment in past years, and you're not expected to have really exceptional artistic skills. It is a challenge to figure out how you want to put this together, and that's good. So you are expected to come up with the plan and put it together and hand it in, and it'll be great. And so I'll see some very different models, and that's absolutely fine. The key thing I'll be looking for is the terms, and then the terms pointing to that part of the embryo. Okay, and just to put the setting in for what's happening in development is that the chick yolk is so gooey that cells divide just on a little patch on the surface of it called the blastodisc, and the blastodisc is a flat circle, and here's an image of it. And what's happening here is that at the end of the primitive streak here, there is a node, a little bump, hence a node in birds, or the node in mammals. And as the node is proliferating, it's sliding along the primitive streak. and covering it up. And so I can draw a little image of that idea. And so here's that blastodisc on the surface, and initially the primitive stalk forms, and a node, a bump forms on this end of it. This is the ventral anterior and posterior end of the animal. And then we start primitive streak regression, which is the equivalent of gastrulation. So lots of stuff is happening. And one thing that's happening is that cells are folding in here. Another thing is happening is cells are proliferating here. And other thing is happening is the node is moving this way. So as the node moves this way, it leaves behind some cells that are covered up, and that includes the cells that are going to make the notochord and the somites and the lateral plate mesoderm. So your model should definitely include those, and the way to see them is to take these little cross sections to see them, like when we cut the embryo open. And so there's a lot happening here, and the embryo starts to develop. And as I mentioned, one thing is that this end of the embryo, the anterior end, the cells are older and further along in development. So if I just cut back to the graph, see up at the anterior end, follow the red dot in the very middle, that's the notochord, just below, just ventral to the blue central nervous system forming as a neural tube. So just following the red down, you can see at the lowest point, you can't see the notochord because that's at the node, and the node is proliferating to produce the notochord and the somites and so forth. And the flanking the notochord are the yellow somites, and you can represent them as being more formed up here and starting to get sliced into individual somites from anterior to posterior. And then flanking the somites are the lateral plate mesoderm that gets hollowed out, and these cells are older, so they're more hollowed out, less hollowed out, and not really hollowed out at all, down at the most down near the node. And so that's the assignment, and I'll talk about it in class 2 and about when the assignment is going to do. Remember that we're building this phylotypic stage of all vertebrates. It's especially true that we and birds, reptiles, mammals, look very, very similar at that phylotypic stage, and we look like what the model is that we're going to try to build. Okay, great. That's it.